Well, good afternoon, everyone. As everyone is getting settled in their seats, we're going to go ahead and get started with our briefing. So we're so happy to see so many of you here for this briefing, which is going to be on diagnostics reform with a special focus on laboratory-developed tests, the FDA final rule, and the VALID Act. This briefing is sponsored by the College of American Pathologists, the CAP. My name is Don Karcher. I am currently the president of the CAP. I'm also a practicing pathologist and professor and immediate past chair of pathology at George Washington University here in Washington, D.C. And I was on service yesterday, so I just had to silence my phone so the resident won't keep texting me about cases. So we're going to get into much more detail in a few minutes, but I just wanted to say a few things to set the table for our discussion. And we have a special guest that I'm going to introduce here in just a moment. So for at least the last 15 years, the FDA, Congress, and multiple stakeholders have worked to develop an appropriate framework for oversight of specific tests called laboratory-developed tests. These tests, LDTs, are developed in a single clinical laboratory for use only in that laboratory, and that's a very important distinction. We're going to go into more detail about the details behind what an LDT is in a moment. These tests are developed to meet a specific clinical need that's not being met by other tests that might be available. These tests over the years have become more and more complex, and that has led to the FDA deciding to regulate them more closely. So today we hope to give you a little bit better idea of what an LDT is, how important they are, not only for laboratories, but particularly for patients, how the FDA final rule that was released recently, what impact that may have on laboratories and particularly on patients, and what Congress can do to create what we think is a better regulatory framework for these tests. Before I say any more, though, I'm very happy and honored to be able to welcome a special guest with us today, Representative Larry Bucshon. Thank you so much, sir, for joining us today. So as you all probably know, Representative Bucshon, and I should say Dr. Bucshon, has been a tireless ally and champion on a long range of healthcare issues. An ally not only for us, but for many in the healthcare industry. With Representative DeGette, he helped lead efforts to create a reasonable and workable framework for oversight of LDTs. Working with stakeholders, Congress, and the FDA, he and his office, with Representative DeGette's office and many other co-sponsors, worked with stakeholders, Congress, and the FDA over a 4-year period to ultimately develop and co-sponsor the VALID Act. He's also been a leader in Medicare payment reform, and so we're very appreciative of all of his hard work and dedication over many years on behalf of medicine and particularly on behalf of patients. So it's my honor to introduce Dr. Larry Bucshon.
Well, thank you. It's great to be here. And thank you for that kind introduction. I've been here a while, so I'm going to put my glasses on to read this. I see it's a pretty young crowd out here. I represent Indiana's 8th Congressional District. It's a rural district in the southwest part of the state. I'm in my 14th year, 7th term, and I did not run for re-election, so I chose to step down. But as was mentioned, over the years, I've worked on a lot of issues related to healthcare, particularly in the provider space, and I want to thank the College of American Pathologists for their support of VALID. I mean, I was a cardiovascular and thoracic surgeon before I came to Congress, and so I did that for 15 years. I also was the president of my medical group, so I've used that broad experience in healthcare to help craft some of the things I've done and I'm doing in Congress. You know, I'm excited to have with us today healthcare providers from the pathology community. And they're going to share with you some background and perspective on a complex FDA issue that Congress needs to address. As most of you know, the FDA is responsible for reviewing diagnostic tests used by healthcare practitioners because there has never been a specific approval pathway established for diagnostics, the FDA now, after recent rulemaking, will use the same framework they are using to approve medical devices. Diana DeGette, my colleague from Colorado, Democrat from Colorado, and I've been talking about this for years, and we've been talking to the FDA for years, and we knew this was coming. That's why we've been working with shareholders to find a better approach. The rule has been widely panned on Capitol Hill in a bipartisan way because it's just not the right approach. And while it may have been appropriate when the medical device framework was created in the 1970s, the field of diagnostics has changed significantly since that time. Not only are tests far more complicated these days, often involving computer programs, they are utilized with extremely more frequency, when before they were fairly uncommon. This is particularly true when it comes to laboratory-developed tests, or LDTs, which are a subset of diagnostic tests that you'll learn more about today. Especially as we move forward toward a future where personalized medicine, genetics, and AI are prominent in medical decision-making, Congress must ensure FDA is equipped to adequately respond. Patients out there are actually counting on us. And the FDA may need more people and more money. That's been one of the criticisms of Valid. I don't disagree with that, so we'll see where that goes. That's the idea between H.R. 2369, legislation I have led for many years with Congresswoman DeGette, which would establish a less burdensome approach to diagnostics review that is best suited to their capabilities. You'll also hear more about that legislation from today's panelists. I'm grateful for to the panel for coming here today and look forward to working with them and my fellow members and senators to ensure FDA works in a way that's best for patients and providers. So I think, actually with the Chevron decision, it may actually give us more of a foothold in Congress to get Congress to I don't want to say understand, but recognize that the rule that has come out of the FDA is not what we should be doing in this space, but we should look at a comprehensive framework, risk-based framework that you'll hear more about here today. That's good for, at the end of the day, good for patients, good for the American people. So it's an honor to be here. Again, I want to thank the College of American Pathologists for their long-term not only support, but input on crafting a legislative framework in this space. With that, thank you very much.
Thank you, Dr. Bhushan, for everything you've done over the years. And we look forward to continuing to work with you. It won't be in Congress, but we'll find a way to work with you. I'll be around. So also, I have another person to introduce today. Also joining me today is a colleague and fellow pathologist, Dr. Joe Saad. Dr. Saad is president of Surgical Pathologists of Dallas. He's also the chair of the College of American Pathologists Council on Government and Professional Affairs. We would have been joined by another pathologist from Louisiana, Dr. Vivek Karcher. Unfortunately, Hurricane Beryl came along, could not get his flight out of Shreveport, Louisiana, and so he was not able to join us today. So we'll just carry on with the two of us. So this is a look at what we're going to be doing after our welcomes, which we just finished. I'm going to get us started with a discussion of what exactly a laboratory-developed test is. So hopefully you'll leave this room knowing much more about that than maybe you know currently. And then also how pathologists and clinical laboratories use these tests every day in our practice, and every day is not an exaggeration. We'll give you some examples. Dr. Saad will then discuss the FDA rule Representative Bhushan mentioned, and its impact on laboratories, on pathologists, and particularly on patients. And that's, at the end of the day, our greatest concern. I'll then discuss actions Congress can take to mitigate fallout from this final rule, and how Congress can create a better way to regulate LDTs, and we think the Valid Act is that better way. We should have plenty of time for questions and answers at the end. So I want to thank you again for being here. We also just want to once again thank Representative Bhushan and DeGette, and also Senator Bennett, who's also been a leader on this issue in Congress. We look forward to continuing to work collaboratively with members of Congress on this issue and many other issues. By the way, the briefing is being recorded and will be available for watching later. And we want to keep this as informal as possible, so when it's time for Q&A, you don't feel obligated to identify yourself if you just want to stand up and ask a question. So with all of that behind us, now let's get started. So laboratory developed tests. So here's the FDA definition. It's kind of legalistic. They are in vitro diagnostic products, that word already sets us off a bit, that are intended for clinical use and designed, manufactured, and used within a single clinical laboratory that is certified under the Clinical Laboratory Improvement Amendments of 1988, or CLIA, and that the laboratory meets the regulatory requirements under CLIA to perform high complexity testing. So let's try to pick apart this definition a bit. First of all, where an LDT is developed is sort of the key of it being a laboratory developed test. It is developed in a laboratory, as the name implies. Sometimes referred to as in-house tests. These can be developed in any type of laboratory, in a community hospital laboratory, an academic medical center laboratory, an independent laboratory, and also by the well-known large laboratory companies that you're all familiar with. So historically, these tests have posed relatively lower risk to patients than commercial test kits. Now, laboratory developed tests are different from most tests that we perform in the laboratory. Most tests are performed using test kits that are purchased from vendors and instruments that are also purchased from vendors. These are different, and traditionally, when these were first being developed a few decades ago, they were much lower risk than commercial test kits. And therefore, they were exempted from FDA oversight. Now, it doesn't mean the FDA didn't feel they had authority to oversee these. Actually, starting in 1976, with the Medical Devices Amendment of the Food, Drug, and Cosmetics Act, which was passed in '76, FDA has chosen to exercise what they call enforcement discretion. They have not enforced the regulatory oversight that they feel that they've had since 1976. So these tests were, simple tests for single analytes. An analyte is something that's measured in a lab test, serum sodium as an example. Or they're tests to diagnose rare diseases in situations where there's not an FDA-approved or cleared test that's available, but the disease needs to be diagnosed. So laboratory-developed tests were developed over the decades in that situation. So since 1976, these tests have become progressively more complex and more commonly developed and run, which prompted the FDA to decide to, question their hands-off approach that existed up to that point. So more recently, these tests have been developed to diagnose infectious diseases, and I'll mention COVID-19 as an obvious recent example, and particularly genomic abnormalities in cancers and other disorders in other clinical scenarios, and we'll give you some examples of those in a few minutes. Now, there have been some very publicized, very egregious, and very high-risk LDTs that the FDA has identified as posing particularly high risk for patients, and we don't argue with that point at all. First of all, non-invasive prenatal screening. This is testing that's done on samples taken from the fetus or from the placenta, and used to screen for Down syndrome and other congenital anomalies in the unborn child. Unfortunately, many of these tests have false positives that have led to unnecessary procedures happening in those pregnancies. Another example is the Oversure ovarian cancer screening test from a few years ago that was marketed and touted as a way of women that may have high risk for ovarian cancer to be screened for that disease. Unfortunately, this test also was plagued with a lot of false positive results, leading to many unnecessary surgeries, removing of ovaries, and other procedures in those women. Now, these are the more egregious examples. Most laboratory-developed tests are not in any way in this category, but these are examples cited as why there needs to be closer oversight of these tests. So, just to give you a little bit more of an on-the-ground, real-world flavor for what these tests are used for, we're going to talk about some examples. Let me start with immunohistochemical stains. Now, these are special stains done on sections of tissue that is done in the laboratory, the stain performed in the laboratory. The pathologist examines the slide, examines the stain, and it assists us in making diagnoses. These tests are performed, these stains are performed in most clinical laboratories in the US, and essentially all pathologists use these stains in making diagnoses. I use them every day. when these stains need to be, new stains need to be developed, there are new proteins that are described on cancer cells, and so we need to develop new stains. Under the current wording in the final rule, these would be subjected to pre-market approval by the FDA before we could use these stains. Just developing a new stain may take a few weeks to a couple of months. This process might extend that to maybe a year or two before we could use those new stains. These are essential in making diagnoses of cancer. And if these stains can't be developed by laboratories, many laboratories have already said that they may stop developing and using these stains, and that means they would have to ship the tissue off to a reference laboratory that may have the stains, wait for those stains slides to come back. Right now, a diagnosis of cancer in my practice can be made in 48 hours. If we had to send these stains out, that diagnosis of cancer may take a week or two to come back, and that we feel is very unfair to patients and very disruptive of patient care. Another similar example is flow cytometry. This is a methodology where proteins are analyzed on the surface or interior of cells, usually cells in suspension. It's used very prominently for diagnosing leukemias and lymphomas. I diagnosed a patient with leukemia just yesterday at GW using flow cytometry. It's a very, very commonly used and very important and powerful technology. Likewise, the final rule has designated this as another test that may, whenever a new protein is described in the literature, it may have to go through full FDA pre-market approval before we can start using it. That might take a year or two or longer even to have that approved. So we think those two are examples of slowing the process, depriving patients of vitally important tests. So I'm going to ask Dr. Saad to talk about these three examples and then talk about the final rule.
Thank you, Dr. Karcher, and good afternoon, everybody. Wuhan 2019, December of 2019, reports emerging of a novel virus, severe respiratory illness, people were dying. A month later, word began to spread that this is probably going to be a pandemic, and then by February, it had arrived in the US, and we were seeing cases on the West Coast. In February, our little lab, which is called PRISM Pathology, started working on a test for SARS-CoV-2, which is the virus that causes COVID-19. By March the 12th, we had the test validated and ready to go, essentially an LDT with caveats. In North Texas, 7 million people, the county health department was able to do 15 to 20 tests a day. There were a couple of labs around the country that could do more, but the waiting period was 2 to 3 weeks. March the 14th, we had a patient in the intensive care unit that was dying from a respiratory illness. We had to fill out a questionnaire in order for the health department to test. He had to have a history of travel, exposure to someone who'd recently traveled, etc. The health department denied the request for the test. I was contacted by one of the infectious disease doctors, and I said, yes, we have the test validated. We can run it in our lab. He said, are you sure? I said, yes, we're sure. We got the sample from the patient. 3 hours later, the test was positive. We called the health department. They said, courier the specimen over immediately. We need to retest the specimen. The specimen went to the health department. The next day, they confirmed that it was positive. That evening, the county judge was on the news saying we have the first confirmed case of community spread of COVID-19 in our community. We're shutting down the city. The health department identified that test, was what he said. It was a laboratory-developed test in our laboratory that identified the first COVID-19 community spread in North Texas. What else have we seen? Zika. Does anyone remember Zika a few years ago, 2014, 2015? We developed an LDT to test for Zika. There was no, and I don't think there is any FDA-approved test for Zika on the market now. Monkeypox 2 years ago. Laboratory-developed tests in our small lab with 3 people, basically, were able to test for monkeypox in our community. Genomic tests for cancer. Representative Bouchon mentioned personalized medicine, and we have been increasingly detecting mutations in certain cancers that are targetable by certain therapies, directed therapies specific to that cancer. Laboratory-developed tests. Think of lung cancer. Think of colon cancer, pancreatic cancer. We are continually discovering new mutations and developing tests that direct the treatment of that patient. Transplant patient testing. Well, the FDA in their final rule did exempt pre-transplant testing, HLA testing, the cross-matching that we do to match a donor and a recipient. But what about post-transplant testing? In order to detect minimal residual disease in the bone marrow. That's essentially an LDT. In our hospital, we do a lot of solid organ transplants, a liver transplant. Now, there's a type of rejection of that transplant. It's called acute graft versus host disease that occurs in recipients of solid organ transplants. Unlike a bone marrow transplant where you want a low-grade chronic graft versus host disease because you want to keep the donor cells, the dominant cells, and suppress the ones that you've transplanted. In organ transplants, you want the opposite to happen. You don't want the donor to attack the host and destroy the host. Acute graft versus host disease, mortality of 80%. The only test is a laboratory-developed test to detect acute graft versus host disease. The FDA final rule would essentially put our little laboratory out of business. We wouldn't be able to offer any of these LDTs going forward. So now to the final rule. As mentioned earlier, the FDA has essentially worked on an oversight framework for almost 15 years, and the CAP has been working alongside the FDA and other stakeholders for almost 15 years on developing an appropriate oversight of laboratory-developed tests. There's been more than one proposal FDA guidance. In 2014, 2015, there was a draft guidance that was disclosed, and several legislative proposals over the years. In 2018, the FDA provided Congress with a draft framework that eventually evolved into the VALID Act. And over a period of 4 years, the CAP and other stakeholders worked closely with Congress in order to modify and improve the VALID Act until 2022, when it reached a point where the CAP and some other stakeholders thought that this was a pretty good framework for laboratory-developed tests. It was passed by the Senate Health Committee in 2022, but unfortunately died later on that year with the MEDUFA, the Medical Device User Reauthorization Fee, in 2022, and so it was stripped from that legislation and never passed. And so that pretty much pushed the FDA to pursue rulemaking. They released the proposed rule in late 23, and then the final rule in 2024, just on May the 6th, it was officially published, the Federal Register, on May the 6th of 2024. However, it regulates laboratory-developed tests under the existing statutory authority, which is the wrong framework to do it. So the FDA did actually make several changes to the proposed rule before finalizing it, including some that the CAP advocated for, but the CAP is still concerned about the burden. Essentially, we agree with the FDA that patient safety is paramount, and we all want to protect patients. However, the rule is written is overly burdensome, it's costly, and we believe it will harm patients by decreasing access to highly effective, well-characterized laboratory-developed tests that are currently on the market now. The FDA actually acknowledged in the final rule that there are many LDTs that are helpful to patients. And so they've provided a pathway for some of them, at least, to remain on the market under enforcement discretion. So they've acknowledged that many LDTs are perfectly fine and safe, and they're allowing them to stay on the market with caveats. The basic provisions under the final rule are that it employs a 3-tiered risk-based structure. You have a Class 1, a Class 2, and a Class 3, the low, moderate, and high-risk categories. But this is a classification for medical devices. And we're really trying to fit a square peg in a round hole here. And there's a phase-out enforcement in 5 stages over 4 years. So it'll be fully implemented by May of 2028. So we have issues with the final rule. It's confusing. The phase-out policy is very confusing and unfamiliar to most of us that work in the laboratory. The FDA has essentially very little role. It has some role in the laboratory currently, but we're regulated under CLIA mostly. And so most pathologists, most laboratorians, laboratory scientists are unfamiliar with working with the FDA. And so we'll need extensive education in order to comply with this rule. And as a result, some laboratories, including our own small laboratory, is considering discontinuing offering laboratory-developed tests. Immunohistochemistry and flow cytometry, many questions. Dr. Karcher discussed what IHC and flow cytometry are. One of the caveats is that if IHC uses automation, then it becomes required to comply with the final rule, at least the phase 1, phase 2, and part of phase 3. If they're currently being used, developing new antibodies, new uses, it'll need to comply with all 5 stages of the final rule. So this is going to be onerous. It's going to increase the time that it'll take laboratories to develop these very important tests. And many laboratories will just not make the effort because it's costly and time-consuming to do it. So there are caveats for allowing unmet clinical needs. There is a category of unmet clinical needs, but there are a lot of caveats in that category, and thus we have many questions. An unmet clinical need is there's no available FDA-approved test that can be performed within a certain time frame. You know, it can be time sensitive. If you have to send it across the country and wait for a week, then that's not for some tests. That's important to have it available more timely than that. So there are many caveats. One is that you need to be within an integrated healthcare system under the same corporate ownership. And so our private lab, which develops LDTs, is on the campus. We're integrated into the health system. We use the same electronic medical record number. We're the same doctors that run the hospital lab and the private lab, but we're not owned by the same corporation. We're a different entity. Furthermore, there's another caveat that the laboratory medical director needs to be employed by the corporation. Well, many of us in private practice and even in academics are independent physicians. We're not employed by the hospital, even though it's our full-time job working in that hospital, 8, 10, 12 hours a day, we're not employed by the hospital. So overall, this will impose significant burdens on clinical laboratories in order to comply. So Congress's response to the final rule was met with general disappointment. Many say that the FDA doesn't have the statutory authority to regulate LDTs under the Medical Device Amendments Act of 1976. Others have said that the FDA just needs to abandon this rule. And many continue to say Congress should work and resume work and double down on work on VALID. And with that, back to Dr. Karcher.
Thanks, Dr. Saad. Those are great examples. That story about COVID is, I've never heard that story. That's fascinating. So you've already heard a good bit about the VALID Act, and we want to give you a little bit more detail on that. This is something that, by the way, this issue is something that we've been working on, we in the College of American Pathologists, for more than a decade, literally about 15 years. And our principles have always been these 3. Any oversight of these tests needs to be balanced. We feel that it needs to be risk-based, and it has to fulfill these following 3 things. Protect patients, and that is absolutely the #1 priority for all of us. It has to provide patient safety. Secondly, it has to ensure continued access to safe and innovative diagnostic tests. Laboratory-developed tests, by their nature, are innovative. They're new things that there's no FDA-approved test for yet, and we want to make sure that whatever regulation does take place, it allows patients to continue to have access to these cutting-edge tests that are safe and accurate. Finally, the framework needs to be the least burdensome possible for pathologists and particularly for laboratories. If it's not, laboratories are going to stop developing these tests. Patients will not have access to them. So why do we feel legislation is needed? FDA oversight of LDTs, we feel, is needed. We believe the FDA is the right agency to oversee these tests. But this oversight has to be appropriate for these tests, and we feel very strongly that the FDA final rule frankly, through no fault of the FDA, because the FDA's hands were tied by the statutory authority that they had dating back to 1976. Unfortunately, what they had to come up with is not appropriate for laboratory-developed tests. This rule regulates these tests as medical devices. These are not devices. This is not something that laboratories market that you can pull off of a shelf and sell to another laboratory. These are tests that are performed by laboratories, overseen by pathologists, and interpreted by pathologists for diagnosing disease. They're not devices. We think intrinsically that's just not appropriate for these tests. The regulation that we currently see in the final rule would result in full FDA oversight, all 5 stages, including pre-market approval for too many laboratory-developed tests. There is a small percentage of laboratory-developed tests, I gave you 2 sort of egregious examples earlier, that really do need full FDA oversight. They can be put patients at risk, but we feel that percentage of LDTs is very small that needs that. Only those highest risk LDTs should be subject to full pre-market approval by the FDA. The FDA oversight of the lower risk tests needs to have significant flexibility so that it allows laboratories, without being overly burdensome, to continue to develop these tests. The goal ultimately is that laboratories will continue to develop LDTs for patients and that patients will continue to have access to these literally life-saving tests. So, the VALID Act. This act, we talked about briefly earlier, that actually it grew out of a framework, draft framework that the FDA provided in 2018 that ultimately evolved into the VALID Act. And the FDA themselves feel very strongly that they need the right framework to oversee these tests. It would establish guardrails on what the FDA can and can't do in terms of its oversight authority, which would lower the burden on laboratories, providing multiple exemptions and new pathways to down-classify tests. In other words, tests that otherwise might be considered high risk would be able to be considered lower risk based on mitigating factors that the VALID Act provides for those tests. It gives the FDA the proper framework with which to oversee LDTs, and I can tell you the FDA feels very strongly that this is the proper framework. They provided the draft framework that ultimately evolved into the VALID Act. It provides the needed flexibility that laboratories must have in order to continue to develop these tests, and that allows us, our laboratories, to continue to innovate and provide access to these innovative tests for patients. This law was developed, and we've talked a little bit about who developed it and how, but it was developed over a 4-year period with broad stakeholder input and bipartisan support. It was introduced last year in the House. It has not so far been reintroduced in the Senate. So what can you do? First of all, we would really like very much for you to go to your member of Congress and consider co-sponsoring H.R. 2369, the VALID Act. We already have mentioned that it is currently sponsored by Representatives Bouchon and DeGette. They've been real champions in this area. It had many other co-sponsors in the past. We would also love to identify a champion on the Republican side. Senator Bennett has indicated an interest in being a champion on the Senate side for VALID. We would love to have a Republican partner for Senator Bennett to ultimately introduce this in the Senate. Also, we would love for you to support efforts to delay the rule. There have been a lot of efforts, and there's a lot of interest in offices to come up with a way of delaying the rule, which would give us all a little more time to develop an appropriate legislative solution. So any support that you want to give for that, we have our staff in the room. If you want more details about VALID or want more details about some method to delay the rule's full implementation while we're working on legislation, our staff would be happy to work with you on that. So that's what we have to present to you. I hope that you have now a little better idea of what we're talking about when we talk about LDTs, some of the issues around these tests. We're happy to answer your questions now. So are there any questions?